The HLA system. So human leukocyte antigen, or HLA, testing is a specialized branch of immunology focusing on human histocompatibility testing. It supports transplantation, transfusion, and immunogenetic specialties. In 1954, Jean Dessaut observed that patients whose sera contained agglutinins had received more blood transfusions than other patients. So the role of HLA. So HLA proteins are part of the adaptive immune system. They are expressed on the surface of cells, and their primary function is to present antigens to T lymphocytes. They facilitate recognition and elimination of foreign antigens. HLA proteins enable self and non-self discrimination. There is a large degree of polymorphism in the major histocompatibility complex, or the MHC. This polymorphism likely evolved to present a wide range of foreign peptide antigens. There are two types of HLA molecules. We have class 1. These present antigens to cytotoxic CD8 plus T lymphocytes. These are found on nucleated cells, dendritic cells, and platelets. Class 2 HLA, these present antigens to the CD4 T lymphocytes. These are found on specialized antigen-presenting cells such as B lymphocytes, macrophages, monocytes, and dendritic cells. They can also be expressed by most cells, including T cells and endothelial cells under inflammatory conditions. For the genetics of HLA, so the HLA genetic region is approximately 4 megabases in size. It is located on the short arm of the chromosome 6. This region is also known as the major histocompatibility complex. For alloantibodies, so they are a barrier to successful transplantation because they can cause rejection of transplanted organs and/or destruction of transfused cells. Alloantibodies arise as a consequence of sensitizing events or exposure to allogenic HLA, such as pregnancy or transfusions. Majority of HLA alloantibodies are IgG, and they are divided into two groups. So those that detect a single HLA gene product and those that detect more than one HLA gene product. There is cross-reactivity. So cross-reactive HLA antigens share structural features but have unique parts. Most cross-reactive antibodies recognize HLA molecules from the same genetic locus. These related antigens are grouped into major cross-reactive groups. Knowing cross-reactivity can help improve donor matching in transplantation and transfusion. For HLA genotyping, so DNA-based methods are used for HLA typing. High resolution, so allele level genotyping is essential, especially for hematopoietic stem cell transplantation. High resolution typing can improve matching accuracy and transplant success. For paternity, so test genetic markers from mother, child, and the alleged father. It can determine if the tested man could be the biological father. It can also be used for disease association, so some genetic markers are linked to susceptibility or resistance to diseases. For platelet transfusions, so HLA class 1 antigens are present on platelets to varying degrees. The alloimmunization to HLA can cause refractoriness to random donor platelet transfusions. Some clinical significance of HLA. So there is transfusion-related acute lung injury, or TRALI. This is an acute lung injury that occurs shortly after transfusion. This is strongly linked to allotypic antibodies in the donor blood. Donor blood is tested for anti-HLA and anti-HNA antibodies to evaluate risk. For hematopoietic stem cell transplantation, so compatibility mainly determined by classical HLA genes. Donor and recipient should match at allele level or high resolution for HLA A, B, C, DRB1, and DQB1 alleles. The umbilical cord blood cells are an alternative when HLA mismatches occur.